and final talk of the session is coming from Jan Soon, who is joining us from Switzerland. Jan is a researcher in the Department of Biology at the University of Freiburg. So whenever you're ready, if you could share your screen, do whatever you got to do, and take it away.
Okay, so can you see my screen well?
Yes, you're all set.
Okay, so thanks a lot to have me here to give a talk. I will just briefly tell you some of my current work regarding the interactions between plant invader and its biocontrol agent in response to climate change. So, I'm conducting an experimental evolution study. So such experimental approach allows me to monitor both phenotypic and genomic changes over subsequent generations, during which I apply the predetermined selective pressure. Like in my case, are two different climatic conditions and two different herbivory pressures. And then combined with the common garden experiment that could allow us better to understand the underlying mechanism of the evolutionary change and also find out the adapted traits. So this is basically the outline of the study. My model species is invasive Ambrosia artemisiifolia and its potential biocontrol agent, Ophrela communa. Ambrosia, probably you guys knows it. It's a native in the US and a part of Mexico, and it's an invasive plant really everywhere in the world. So it's a big problem for agriculture, and also it's highly allergenic pollen. It's spread like like many other invaders along the traffic roads or disturbed area. So in Europe, it's a really not very welcome plant, because the allergy problem, and also cause the problem for the tourist issue. Then the potential Ophrela communa, this flea beetle, accidentally arrived in Europe about 2013. First discovered in North Italy and South Switzerland. And last year we already observed in Romania. So it's about 100 kilometers per year to spread towards east of Europe. And in Europe, this beetle actually did pretty well job so it's not really classic biocontrol, it just accidentally introduced, but it still did very much damage on Ambrosia. This is a maize field in Italy, North Italy. And you see for the spring sown crops, they harvest about June. After that, for such a high nutrient crop field, all this dry shoot actually are ragweed. And in August, you can see they are all completely eaten up by Ophrela. In China, I think it's a really successful biological control, classic biological control agent that's got mass release and controlled ragweed, at least in south part of China, very well. You can see one outcome in North Italy of this beetle is reduction of ragweed pollen about 80%. So there's recently a lot of study about this beetle, the risk assessment, if this could be potentially a successful biocontrol agent in Europe against invasive plant. So this is my field cages for my experiment in North Italy. I have 22 x 2 x 2 meter cages with 2 levels full factor design. So 2 different temperature schemes and 2 different herbivory treatment. That is with or without. And I did this experiment over 4 generations. So this is the yellow star is where my cages are, and then the initial 20 identical population coming from 60 maternal families that from the 19 population of the purple point around this site. So we want to create quite of a natural population, but still have a high genetic diversity within the population. And North Italy was already shown to have really high within population genetic diversity. So we could have a quite good chance to have this high potential evolvability. Then I did a pilot experiment to examine the maternal families that I used in this experiment. So giving examples for days or flowers, you can see each color line represents one maternal family, and you can see that there is a significant variation in both trait means and also the plasticity among the parental generation. So this experiment allows us to both compare the result cross time, so like parents and offsprings across generation, that's allochronic comparison, and also do the comparison for the offspring that coming from a different treatment, or combine the population genetic data with the phenotypic data that come from the common environment. So the first result I want to show you is the sequence data. Here is the sequence composition that compare all the polymorphic sites from the plant that from the different treatment and generation. So each point here is one population. So gray is the parents, and the red means the offspring from the worming treatment, and the blue means offspring from the ambient temperature treatment. Without this black circle means with or without Ophrila treatment. And the darker color means the F4 offspring, and the lighter is F1 offspring. Okay, so you can see clearly from genetic data, the worming treatment has much greater genetic divergence from control on the offspring population than the herbivory pressures. I also calculated the selection coefficient of every locus. So this the positive value means the increased allele frequency, and negative means decrease. Also, based on the 5 replicates, I could statistically calculate the selection probability. In this way, I can annotate the one has probability more than 90%, which given here by the red circle, red points here. So in general, these are all the SNPs that under selection. You can see there's still some locus that also show differences for control cages or populations. So our treatment allows us to annotate everything out of the control, because it's probably just the effect of the cage. So the rest, there's the not overlap part are the things we are interested. And for the briefer preliminary result of the annotation, we already found indeed there are some genes directly respond to heat or to growth, which might be very important. Now move to the common environment that I took the seeds from the both parental generation and the seeds of the offspring from the field and grow them in the common environment. And what we found for the F1 offspring, there is a showing that plant from worming cages flower later and grow bigger as compared to control and the parental generation. So this such a observed evolutionary change probably due to selection by differential mortality, growth, and reproduction among the initial sown genotypes. They also calculate the quantitative genetic variation. You can see in common environment, offspring from worming population, the red point showed much more convergent phenotypes in 7 out of 9 plant traits. Then I was wondering, does this, the 2 traits that showed high different phenotypic differences also meaning they were selected by the worming treatment? That's what we do. We compare the phenotypic and the genetic divergence cross-generation for different treatment. So this just using the Hadlon matrix to calculate the evolutionary speed. For this way, we assume if this PG ratio higher for worming than the control population, that meaning this trait probably under selection due to worming treatment. And indeed, at the end, we found these 2 traits, biomass and days to flower, showed that there's a higher PG ratio for worming than control, meaning there's a greater response to selection under worming condition. Now let's move to the second experiment that we did to compare the growth and defense. You see, this is what I always observe in the cage. Some individual plant really damaged very well, but just the neighbor plant still looks so happy and healthy. This experiment will also compare the cross-generation, but besides genetic, we also compared the chemical profile and combined with common garden experiment. So For genomic, it's what I said before, only the worming showed effect, but metabolomic profile, which is very different case. So basically, besides generation differences, the mainly differences you can see here is there's significant separation between the beetle and non-beetle offspring. Here, the worming showed really no effect of the chemical profile. In the common environment, we first tested the Ambrosia performance. Oh, here I already used the F2 generations. So it's very similar to the F1 generation that the offspring plant from the worming cages, they flower later, grow bigger. And the beetle treatment has not really very strong effect. But for biomass, for example, we do see there is interaction between beetle and worming. And then we did test the bioassay for the resistance of Ambrosia. So we did both preference experiment in the insect rearing cage by release a cup of beetle to the 4 plant that coming from 4 different field treatment. And then also did a performance experiment in the petri dishes, just release a larvae on the different Ambrosia leaves. So you can see basically it's really in the same line as the chemical profile I just showed you, that the Ophrella definitely prefer to lay eggs on the plant that has no beetle cages, from the no beetle cages. And also from this no beetle cages, they have much less mortality. That means this offspring Ambrosia from beetle cages has much higher resistance as compared to the plant from non-beetle cages. If we combined growth and defense, basically we found that there is in general, in both ambient and elevated temperature, we found a positive growth defense correlation. There are many theories to explain that. For example, someone assumed that within the genotype is probably there is a trade-off, but in general, for the species level, you find positive correlation. Or there's another assumption saying for the species, each point here is a species. For the species that experienced physiological stress, they might showing trade-off between growth defense. But for the species has no limit for the resource to growth, they might showing positive for growth defense. So for specific Ambrosia artemisiifolia, previous studies already showed from both native and introduced ranges. In general, there's a positive growth defense relationship. So our finding is quite in line with previous study. But what we found different is in the ambient temperature, we found the increased resistance may be costly, definitely different from the elevated temperature. So this interaction, meaning that the worming probably will decrease the biological control efficacy. Now I already show you that there's a genetic and a phenotypic changes of the Ambrosia that from among all the different treatment. And then I was logically wondering, does this also will alter the soil microbial or the abiotic properties? For this, after 4 generations of this field evolutionary experiment, we then collect the soil from the experimental soil from the cage, within the cage, and also some soil outside the cage that never have experienced Ambrosia as an additional control. Moreover, we then examined both abiotic property and the biotic community. And then we did the plant soil feedback in the common environment using the offspring seeds that we collected from the field after 4 generations. So this experiment, we again combined both field data and the common environment data. First, the abiotic. So the gray color here, meaning the soil we collected outside the cage that never have the Ambrosia experienced. And other color means from the different treatment. So you can see for abiotic properties, there is clearly differences between the within and outside the cage. There's many traits differ, but the one of things I definitely noticed the pH. The pH definitely increased for when they experienced the Ambrosia. But for the biotic, we measured both about bacteria and fungi community. first, again, outside cage, the soil from outside cage showed quite different. But the interesting thing is both bacteria and fungi also significant effect affected by our different treatment. And seems like the treatment definitely has effect because you see the similar color. So the from lighter to darker, meaning my 5 replicates. So the similar color like bluish, greenish, reddish, they are also clustered. So our field treatment somehow directly or indirectly through the Ambrosia composition change that affect the community, soil microbial community somehow. So I don't know yet is direct or indirect change. We are still trying to explore this, but given the initial population, experimental population are really identical, just 4 generations, we already see the differences. I'm still quite surprised for that. Then just quickly show you one interesting result from the plant soil feedback experiment that just we have different plant source and soil source and then different combination. There are one very interesting result that you can see the blue dots here showed always a negative. So the soil from the beetle cages showed a negative feedback to the growth of ambrosia, also to the beetle cages. This is like quite like opposite from the worming cages that has basically no negative feedback. Just quickly sum up the whole study. I showed you that selection by worming and beetle changed the genetic or metabolomic composition of ambrosia population with consequences for their offspring phenotypes. Specifically, beetles selected for increased defense with no cost only under worming. And selection by worming and beetle also changed the soil microbial composition, resulting in different plant soil feedbacks among offspring. So under climate warming, biocontrol may result in larger offspring ambrosia, but with increased resistance and complex plant soil feedback, which all might decrease the biocontrol efficacy over time. So thanks a lot for your patience, and I'm waiting for the question.
Great, thank you so much. And what an impressive amount of experimentation you showed us. That's really, really great. So any questions? We have, a couple minutes before our break, so we'll give people some time to type anything in the Q&A. It's too bad. These talks are great and so informative, but it's, I'd love to hear that message of, oh, warming makes biocontrol work better and herbicides are going to be more efficient. And, it's obviously not reality, but yeah. So any questions, anybody? Okay, we've got one here. So Carrie asks, have you heard of other mismatches of biocontrol and target plants under climate change? So are you familiar of any other examples that kind of parallel what you found?
I think if I got it correctly, but I really have to double check the reference. I remember there's one study about alligator weed. I think there's one paper exam that when they are experience the warming, the biocontrol agent will have at least at experimental cages, they might eat a little bit more the native species, similar species, when compared to there's no warming time. So there might be possible risk for the host specificity. Like you examined this biocontrol agent is very well, but then under warming, then the host specificity may be changed a little bit. I remember this about alligator weed. There is one paper published on Global Change Biology or somewhere, or Ecology Letters. I could, if anyone interested, I could double check the paper.
Could I pipe in?
Sure.
There's a cereal leaf beetle is a huge pest in the United States under amazing biocontrol out west. And as climate change has come on in the last 10 years, it started having refugia in the spring based on the difference in the phenology of the host and the pest, and biocontrol efficacy is going down because of warming temperatures in the spring. So.
Thanks, Julie. Okay, Jan, if you wouldn't mind stopping your screen share, and we'll just wrap up real quick.
Okay.
So thank you so much to our speakers. We really appreciate you guys being here and sharing the amazing science you've been doing. And thanks everyone for attending. This is the last session of talks